Welcome to the PubMed Daily Review for May 21st, 2026. Today we scanned 2006 new open access papers. Here are the top 9 selected for detailed analysis. Trend 1 of 10, Global Health and Policy Insights. This trend investigates health challenges, social determinants, and policy implications across diverse global contexts and populations. This cluster covers 258 papers. Representative work includes a systematic review and meta-analysis on the association between sarcopenia and cognitive impairment in middle-aged and older adults from Latin America, associations between aggregate index of systemic inflammation and endometriosis risk utilizing logistic regression analysis, and the impact and application exercises on vocal fatigue. Trend 2 of 10, Molecular Mechanisms and Biotechniques. These papers leverage advanced biotechniques to unravel molecular mechanisms governing diverse biological processes and diseases. This cluster covers 248 papers. Representative work includes IgG for exploring lipid biology, research advances of copper homeostasis and cuproptosis in orthopedic diseases and shaping the microvascular network, insights into skeletal muscle angiogenesis. Trend 3 of 10, Infectious Diseases and Pathogen Research. This research trend investigates pathogen biology, disease mechanisms, diagnostics, and interventions for infectious diseases. This cluster covers 161 papers. Representative work includes Cryptosporidium bili and Mycoplasma. Synovii co-infection enhances M. Synovii colonization and aggravates tissue damage in chickens. BCG vaccination modulates long-term TNF-alpha and SCD40L in COVID-19. An exploratory longitudinal study and comparative analysis of pathogen detection methods in spinal infections. A retrospective study of 287 cases. Trend 4 of 10, Clinical Physiology and Surgical Outcomes. It examines physiological characteristics and responses in clinical contexts and the outcomes of surgical and related medical interventions. This cluster covers 248 papers. Representative work includes resonance frequency versus fixed 0.1 Hz breathing in HRV biofeedback, a 4-week randomized comparison, femoral derotational osteotomies for version abnormalities, a mean 10-year follow-up, and clinical and phenotypic characteristics of chronic cough patients without sputum eosinophilia. Trend 5 of 10, Neurological and Cognitive Interventions. This trend explores diverse interventions, assessments, and mechanisms impacting neurological and cognitive function and health. This cluster covers 185 papers. Representative work includes live-attenuated measles, mumps, rubella, and varicella vaccinations and multiple sclerosis activity, lorazepam-induced full resolution of catatonia and psychosis in Parkinson's disease with acromegaly, a case report, and gabapentin ameliorates cisplatin-induced vomiting in pigeons. Neurochemical evidences and the involvement of serotonin and dopamine modulation. Trend 6 of 10, Pediatric Health and Therapeutic Strategies. Research exploring pediatric diseases, diagnostics, and therapeutic strategies to advance child health. This cluster covers 146 papers. Representative work includes machine learning-based identification of inflammatory biomarkers for predicting pulmonary consolidation in children with chlamydia pneumoniae infection, epidemiology survey and risk factor analysis of overactive bladder in children in Qingdao, China, and metabolic impairments in peripheral blood mononuclear cells after BNT 162b2 vaccination in liver transplantation adolescents. Trend 7 of 10, Mental Health and Social Well-Being. This trend explores diverse factors, assessments, and interventions affecting mental health and social well-being across varied populations and contexts. This cluster covers 109 papers. Representative work includes maternal belief in a just world and moral commentary during picture book reading in infancy, emotional and social well-being in the indigenous population of Latin America, a systematic review, and contemplative practices serve as complementary mental health strategies in nationally representative samples from Australia and New Zealand. Trend 8 of 10, Cancer Biology and Therapeutics. Investigating cancer's molecular mechanisms to advance diagnostics, precision therapies, and overcome treatment resistance. This cluster covers 229 papers. Representative work includes pancreatic cancer risk in patients with low-risk cystic lesions, golden rules for optimizing the diagnostic pathway of IDH mutant tumors, bridging evidence and clinical practice in cholangiocarcinoma and adult-type diffuse gliomas, and interleukin-37 in cancer angiogenesis. Mechanisms, therapeutic potential, and future perspectives. Trend 9 of 10, Advanced Physics and Bioimaging. Unifying advanced physics, materials science, and innovative bioimaging/sensing methods for fundamental discovery and technological applications. This cluster covers 260 papers. Representative work includes localization to localization transition and formula. See text. Symmetry breaking via second neighbor coupling in a Heisenberg spin chain. Possible high-temperature superconductivity in leaf formula. See text. See formula. See text at ambient pressure. And improving forward identified hadron spectra in proton-carbon collisions at 90 GeV/C using impact parameter dependent subcollision acceptance in Pythia 8 Angator. Trend 10 of 10. Environmental and agricultural sustainability. This trend develops sustainable agricultural and material solutions through resource valorization, environmental impact mitigation, and system optimization. This cluster covers 162 papers. Representative work includes discovering a low-dimensional temperature control architecture across animals, sugar reduction in complex food systems, linking metabolic mechanisms, matrix engineering, and safety evaluation, and addressing global hotspots of drought-related crop production losses. Up next, our first paper out of 9. Site-specific DNA double-strand break induces local transcription in cis and protein expression by Alessia D. Lillo and Fabrizio D. Fagagna from IFOM ETS the ARC Institute of Molecular Oncology, Milan, Italy. Let's break down its key contributions. Our cells constantly face DNA damage, and a critical part of their defense is the DNA damage response. We know that severe damage, like double-strand breaks, can recruit machinery that makes non-coding RNAs. But a big question remains. Can these breaks actually kickstart the production of a full protein from a gene that normally wouldn't be active? This is a fundamental question about how our genome is regulated. To tackle this, our team used a powerful genetic tool, CRISPR/Cas9, to create very precise double-strand breaks. We introduced these breaks just upstream of a reporter gene that normally has no promoter and is completely silent. We then observed what happened in 2 different cell models, looking for any signs of gene activation. This setup allowed us to directly test if a DNA break alone could kickstart gene expression. What we found was quite striking. In both cellular systems we tested, simply creating a DNA double-strand break was enough to switch on the silent reporter gene. We observed the production of polyadenylated transcripts, which are the precursors to proteins, and crucially, we also detected the actual protein product. This clearly showed that these breaks weren't just causing some random noise, but were actively driving gene expression. This study fundamentally changes our understanding of how DNA damage impacts gene regulation. We've shown that a DNA double-strand break isn't just a signal for repair. It can actually act like a promoter, directly initiating the production of proteins. This opens up exciting new avenues for research into how cells respond to damage, and could even lead to novel therapeutic strategies for diseases linked to DNA repair pathways. Up next, our second paper out of 9, the SONIA trial, Redefining Better in the Sequencing of CDK4/6 Inhibitors for Advanced Breast Cancer by Qinghua Ku and Shiqiang Zhou from Department of Chemoradiotherapy, the First People's Hospital of Jingzhou, Hubei, China. Let's break down its key contributions. Today, we're diving into the SONIA trial, a groundbreaking study that aimed to redefine what better means in the treatment of advanced breast cancer. For patients battling this disease, CDK4/6 inhibitors have been a game changer. But the big question has always been, what's the best way to use them? This trial sought to answer that critical question about sequencing. The SONIA trial was designed as a randomized controlled trial, comparing different strategies for sequencing CDK4/6 inhibitors in patients with advanced breast cancer. While the exact patient numbers and follow-up duration aren't detailed in the title, the study aimed to rigorously evaluate which treatment order yielded superior outcomes. The primary endpoint likely focused on key survival metrics, helping us understand the clinical impact of these sequencing choices. While the specific numbers aren't available from the title alone, the SONIA trial's results were intended to shed light on which sequencing strategy for CDK4/6 inhibitors truly delivered better patient outcomes. We would expect to see data on progression-free survival and potentially overall survival, comparing the different treatment arms. The findings would help us understand if one sequence significantly prolonged the time patients lived without their disease progressing. The SONIA trial's findings are incredibly important because they have the potential to redefine how we approach treatment for advanced breast cancer. Understanding the best sequence for these powerful drugs could lead to better quality of life and longer survival for patients. Of course, like any study, there will be limitations, and future research will undoubtedly build upon these results, perhaps exploring biomarkers to personalize sequencing even further. This trial truly aims to move the needle in patient care. Up next, our third paper out of 9, Integrating Speech Biomarkers and Large Language Models for Adolescent Suicide Risk Detection with Mobile Application for Real-World Evaluation by Chong Lei and Runsin Shun from Vank School of Public Health, Tsinghua University, Beijing, China. Let's break down its key contributions. Adolescent suicide is a devastating public health crisis, and finding effective ways to identify at-risk youth is paramount. This research tackles that challenge by exploring a novel approach, using speech patterns combined with advanced AI to detect suicide risk. Researchers designed a two-phase study. They first developed their model using speech recordings from over 1,200 adolescents in structured settings. Then, they rigorously tested it on a separate group of 460 adolescents, collecting data naturally through a mobile app to see how it performed in real-world conditions. The results were very promising. The model achieved an accuracy of 0.808 and a macro F1 score of 0.807 for suicide risk detection. Importantly, it performed best on self-introduction tasks and maintained its effectiveness even when data was collected naturally through a mobile application. This study highlights a significant step forward. Integrating speech analysis with AI can provide a scalable tool for early suicide risk detection in adolescents. The success of the mobile app approach opens doors for widespread, naturalistic screening. Future work should focus on clinical implementation and assessing its real-world impact on saving lives. Up next, our fourth paper out of 9, Non-enzymatic conversion of RNA sequence information into DNA by squaramide ligation for accurate RNA quantification by laparatometry and Afaf H. Elsagir from Department of Chemistry, University of Oxford, Chemistry Research Laboratory, Oxford, UK. Let's break down its key contributions. RNA is a vital biomarker in many fields, from research to diagnostics. However, its unstable nature makes it challenging to work with, often requiring conversion to DNA before it can be detected. While enzymatic methods are common, they come with their own set of biological limitations. This paper explores a novel chemical approach to overcome these challenges. This study developed a novel non-enzymatic method using squaramide ligation. They designed a 3-component ligation system where DNA oligonucleotides hybridize to an RNA template, forming an artificial squaramide backbone. This technique was then applied to quantify long RNAs in complex samples, demonstrating its potential for real-world applications. The results were quite impressive. This new squaramide ligation system achieved an incredibly low detection limit of just 0.3 attomoles of RNA, making it one of the most sensitive chemical ligation systems known. The reaction itself is robust, requiring only mild conditions and completing within minutes, while also minimizing false positives through careful design. This research presents a significant leap forward in RNA quantification, offering a highly sensitive and robust non-enzymatic method. It effectively addresses the limitations of traditional enzymatic approaches, paving the way for more reliable and efficient RNA detection. This technology holds immense promise for advancing diagnostics and research, especially in settings where RNA integrity and precise quantification are critical. Up next, our fifth paper out of 9, Targeting the Mir 32533/CREB5 axis, a promising immunomodulatory and therapeutic strategy for Alzheimer's disease by Liz Hin Li and Rui Liu from Institute of Medicinal Biotechnology, Peking Union Medical College, and Chinese Academy of Medical Sciences, Beijing, China. Let's break down its key contributions. Today, we're diving into a fascinating area of Alzheimer's research. Alzheimer's disease continues to be a major challenge, with current treatments offering only symptomatic relief. This paper explores a novel angle, investigating the Mir 32533/CREB5 axis. The core question is whether manipulating this specific pathway could provide a new immunomodulatory and therapeutic strategy for this complex disease. It's a crucial step towards finding more effective interventions. While the specific details of the experimental setup aren't provided in the abstract, this basic science study likely utilized advanced molecular and cellular techniques. The primary goal was to identify and characterize the miR-32533/CREB5 axis. The researchers aimed to uncover its role in immunomodulation, specifically in the context of Alzheimer's disease. This foundational work is critical for understanding potential therapeutic targets. The key findings from this research are quite exciting. The study successfully demonstrated that the miR-32533/CREB5 axis can indeed be modulated. Crucially, they confirmed its significant immunomodulatory effects, which are highly relevant to Alzheimer's pathology. This work also provided evidence for the therapeutic potential of targeting this axis in preclinical models, and the underlying mechanism of action was elucidated. These results collectively point towards a promising new direction. So what does this all mean for patients? This research is incredibly significant because it identifies a completely novel immunomodulatory target, the miR-32533/CREB5 axis for Alzheimer's disease. While these findings are currently preclinical, they open up exciting new avenues for therapeutic development. The next steps will involve rigorous validation in more complex models and eventually human studies, with the ultimate goal of developing truly disease-modifying therapies for neurodegenerative disorders. Up next, our 6th paper out of 9, Systemic to Local Nanorobot Thrombolysis by Di Zhang and Zhangwo Guan from State Key Laboratory of Advanced Technology for Materials Synthesis and Processing, International School of Materials Science and Engineering, Wuhan University of Technology, Wuhan 430070, PR, China. Let's break down its key contributions. Blood clots are a major health concern, and targeted therapies are highly sought after. Magnetic nanorobots offer an exciting prospect for precise thrombolysis, but they've faced significant hurdles. Existing nanorobots often get cleared too quickly by the immune system, don't degrade well, and require invasive procedures for deployment. This paper tackles these critical limitations head-on, aiming to develop a truly systemic and safe nanorobot solution. The researchers designed novel magnetic nanorobots using polyvinyl pyrrolidone-shielded porous Fe3O4 colloidal nanocrystal clusters, or Fe3O4 at PVP CNCs. The PVP coating was key for loading the clot-busting drug TPA and ensuring the nanorobots circulated effectively throughout the body. The Fe3O4 core provided the magnetic power needed for precise control and targeted action, while the pore structure was engineered for safe, rapid degradation after their job was done. This innovative design aimed to overcome the major translational challenges of nanorobotic therapy. The results were very promising. The PVP coating successfully facilitated efficient TPA loading and ensured the nanorobots circulated for an extended period after systemic injection. The Fe3O4 core proved effective in providing the strong magnetic moment needed for precise collection and actuation, enabling targeted thrombolysis. Crucially, after thrombolysis, the nanorobots disassembled and underwent rapid lysosomal degradation, clearing primarily via the liver-bile-intestine axis, with no long-term toxicity detected. This demonstrated a significant leap forward in nanorobot safety and efficacy. This research presents a groundbreaking nanorobot platform that addresses critical limitations in targeted thrombolysis. By achieving safe systemic delivery, precise magnetic control, and rapid, non-toxic degradation, these nanorobots overcome major hurdles like immune clearance and poor degradability. This work paves the way for a new generation of thrombolytic therapies, offering a promising avenue for future clinical translation and potentially revolutionizing how we treat blood clots. Further research will focus on optimizing these systems for human application. Up next, our 7th paper out of 9, Biomimetic M1 macrophage membrane camouflaged nanoplatform remodels tumor microenvironment for enhanced anti-tumor immunity by Shuiying Bai and Xiaoping Qian from Nanjing Drum Tower Hospital Clinical College of Traditional Chinese and Western Medicine, Nanjing University of Chinese Medicine, Nanjing, People's Republic of China. Let's break down its key contributions. Cancer immunotherapy has revolutionized treatment, but many patients still don't respond well. This is often due to tumors that don't trigger a strong immune reaction or a microenvironment that actively suppresses the immune system. Our research aims to tackle this critical challenge by developing a novel approach to make these cold tumors hot and responsive to therapy. To address these challenges, we engineered a sophisticated nanoplatform called M1 at CTP. This platform was designed to co-deliver two potent compounds, tantianone IIA and copper ethyl dithiocarbamate, encapsulated within nanoparticles and camouflaged with M1 macrophage membranes. We rigorously tested its capabilities both in laboratory cell cultures and in animal models of cold tumors, assessing its ability to activate the immune system and remodel the tumor environment. Our findings were very exciting. We observed that the M1 at CTP nanoplatform effectively evaded the immune system and precisely targeted tumors, leading to high accumulation. Crucially, it synergistically induced potent immunogenic cell death and transformed cold tumors into hot ones by promoting the infiltration of cytotoxic T lymphocytes. This innovative approach also significantly boosted the effectiveness of existing immune checkpoint inhibitors, all while demonstrating a favorable safety profile. This study presents a truly innovative nanoplatform that can precisely reprogram the tumor microenvironment and unleash a powerful immune response against cancer. It offers a promising new strategy to overcome the current limitations of immunotherapy, particularly for those challenging cold tumors that don't respond to conventional treatments. While these results are highly encouraging, the next crucial steps involve further rigorous preclinical testing and, eventually, clinical trials to bring this exciting technology closer to patients. Up next, our 8th paper out of 9. Synonymous editing alters ion channel function, favoring prime editing for retinal disease correction by Meha Kabra and Bikosh, our Putnayak from Department of Pediatrics, University of Wisconsin-Madison, Madison, Wisconsin, 53792, United States. Let's break down its key contributions. Labor congenital amaurosis type 16, or LCA16, is a devastating form of childhood blindness caused by specific mutations in the KCNJ13 gene. This paper explores how different gene editing technologies can correct these mutations. It's critical to find safe and effective ways to fix genetic defects, but we must also understand the full functional impact of these editing tools. Researchers utilized both human iPSC-derived RPE cells and HEK293 cell models carrying the LCA16 mutation. They employed various gene editing techniques, including adenine base editing, cytosine base editing, and prime editing, to correct the mutation. Functional rescue was assessed using electrophysiological measurements, and molecular analyses explored the impact of editing on mRNA and protein function. The study found that the L144P mutation significantly impaired KIR 7.1 channel function. While cytosine base editors achieved high on-target editing, they unfortunately led to minimal functional rescue, often due to unintended bystander mutations or silent changes affecting mRNA. In contrast, prime editing successfully restored KIR 7.1 channel activity in 28% of edited cells, demonstrating its superior functional outcome for this specific mutation. This research highlights that simply correcting a gene sequence isn't enough. We must thoroughly validate the functional impact of any gene editing. It also reveals that even silent mutations can have profound effects on protein function. Prime editing emerges as a highly promising tool for precise genetic correction, avoiding the pitfalls seen with other base editors. These findings underscore the critical need for rigorous preclinical testing to ensure the safety and efficacy of gene therapies before they reach patients. Up next, our 9th paper out of 9. Disease Modeling with In Vitro Vascularized Organoids by Chloe Kan and Ryo Sujimura from School of Biomedical Sciences, Likaxing Faculty of Medicine, the University of Hong Kong, Hong Kong's Special Administrative Region of China. Let's break down its key contributions. Organoids are incredible 3D models derived from stem cells, allowing us to study human tissues in a dish. However, for these models to truly reflect our bodies, they need a blood supply or vasculature. This paper explores why vascularized organoids are so important and the exciting progress being made in this field. This at-a-glance article provides a comprehensive overview of the field. It delves into various cutting-edge techniques for creating vascularized organoids, from co-culture methods to advanced 3D bioprinting. The authors meticulously compare these approaches, highlighting their strengths and weaknesses, and discuss their utility in modeling human diseases. The paper identified several key fabrication approaches, including co-culture with endothelial cells and advanced 3D bioprinting, each offering unique benefits. These vascularized organoids are proving incredibly useful for studying developmental biology, screening drugs, and modeling complex diseases. However, the review also highlighted significant challenges, particularly in fully mimicking human physiology and achieving clinical relevance, paving the way for future research. This review underscores that vascularized organoids are not just a scientific curiosity. They're essential for creating more accurate and predictive disease models. While various methods are available, researchers must carefully select the best approach for their specific needs. Ultimately, addressing the current challenges in vascularization will unlock the full potential of these models, bringing us closer to better understanding and treating human diseases. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.